All right, welcome back for more proteomics. So at this point, we've had a good discussion on how amino acids are incorporated together through forming peptide bonds to form a polypeptide. We've talked about specific amino acids and their side chains and their chemical properties. And then in the last video, we talked about we gave a quick little review on how proteins are made in the cell. Just quick little review here. The ribosome is going to read a messenger RNA molecule, and that messenger RNA molecule will provide the ribosome instructions on which amino acids to put in which position in the polypeptide. So once the ribosome has reached its stop codon and it releases the polypeptide we don't consider it a protein just yet. It's not going to actually be a functional protein until it folds into its final and most stable three-dimensional shape. And that is going to be the topic of this video here. We are going to talk about the hierarchical way in which proteins fold into their final functional shape. So let's go ahead and get after this. So it's going to be convenient for us to think of protein folding as a stepwise process. So if you look at the figure over here on the right, we can think of it as kind of a 3 or 4 step process. At this point in our discussion, we are familiar with what we would call the primary structure of the protein. The primary structure just refers to the sequence of amino acids starting with the N-terminus and ending with the C-terminus. So from start to finish, which amino acids go in which position. We're already familiar at this point that most commonly the first amino acid is going to be methionine because that is the amino acid coded for by the start codon. And then whichever amino acids come after that in which order, that's just going to depend on what the code is in the messenger RNA. And then as you go down the list here, you'll see that the primary structure, which just kind of looks like a string of pearls here, it is eventually going to fold and undulate in a number of different ways until we get our final three-dimensional shape. And this is highly significant because it is ultimately going to be the shape of the protein that is going to determine what that protein's function is. Now, it is true that the primary structure is ultimately what gives us the final three-dimensional shape, but it's the shape that ultimately is what is important, as we will see examples of later. So the 4 different levels of protein folding are called the primary structure, which we just talked about, the secondary structure, which we will have a good discussion on, tertiary structure, and then finally quaternary structure. So we're going to address each of these one by one and learn a little bit about how they kind of fit into this stepwise process of getting a protein folded. So as we said, the primary structure of a protein, it's literally just information, right? It literally is just how many amino acids are in the protein and in what order do they come in. As we said, this is determined by the nucleotide sequence in both the mRNA and in the gene found on whichever particular chromosome that gene was found. So the primary structure is represented by a 1 or 3 letter code for each amino acid written one after another, starting with the N-terminus and then leading up to the C-terminus. So this is why we mentioned in the first video that at some point, you'll want to obviously take your time with doing this because it's a little bit like learning a new language. But at some point, you'll want to familiarize yourself with both the 1 and the 3 letter codes for amino acids, because at times during the semester, especially when we do bioinformatics stuff in class, we're going to be looking at primary sequences of proteins, and it's going to be listed as M, W, D, A, G, C, and so on. So obviously, you're going to want to know which amino acid is which. So something that you can kind of take your time with, but you definitely want to eventually learn that. The sooner the better. So the analogy I think I invoked on the previous slide is that you can kind of think of the primary structure as kind of like a string of pearls. Just imagine each pearl on the string being a particular amino acid, and the string being the peptide bonds that hold each amino acid together. So the primary structure of the protein is literally just imagine stretching the whole polypeptide out into a long linear strand. So All the amino acids here are obviously present and accounted for, but the polypeptide is obviously stretched out like this, and it's not going to have any function because it has not folded at all yet. And as we said on the previous slide, a protein has to be folded if it's going to have any function, regardless of what kind of protein we're dealing with here, whether it's an enzyme or whether it's some type of transporter protein, or maybe whether it's a transcription factor. Whatever its job is in the cell, it's not going to be able to do that job at all if it looks like this. It has to be able to fold into a particular three-dimensional shape. But the interesting thing is, the primary structure is all the information that is needed to ultimately determine the three-dimensional shape of the protein. But, like I said, it does not actually itself have any activity or function. Okay, so now let's talk about secondary structures. So this is going to be kind of the next step towards getting the protein folded. As we said, proteins have to attain some type of three-dimensional structure in order to have any kind of function. The first step in getting there is to come up with a number of different secondary structures. This is going to involve non-contiguous, meaning amino acids that are not right next to each other, they're not peptide bonded to each other, interacting with each other, as we are about to see. So there are two major types of secondary structures. The first one that we're looking at here on the left, and we'll introduce more on the next slide, is called an alpha helix. Regardless of whether we're talking about the alpha helix or the similar beta sheet, the two major types of secondary structures, these structures are stabilized and held together by hydrogen bonds. And hopefully you remember that from your chemistry and your introductory biology classes. Hydrogen bonds are the result of weak electrostatic attractions between highly electronegative atoms like oxygen and nitrogen, and then hydrogen atoms, which are not electronegative at all, having attractions for those oxygens or for those nitrogens. So for secondary structures, the thing that you'll notice here is that the hydrogen bonds, which are represented by dotted lines here, the hydrogen bonding is between the backbone of the polypeptide, attracting atoms in the backbone of the polypeptide somewhere else. So you'll notice that represented here in this alpha helix, you do not see any of the side chains present, and that is because the side chains do not participate in the formation of secondary structures. The side chains are going to be a lot more heavily involved in stabilizing tertiary structure, and then also allowing the protein to interact with solvent and to stabilize the final structure of the protein. So side chains are not really so much involved here. Secondary structures are formed via the interaction between parts of the actual backbone. So what you're seeing here is the alpha carboxyl group. This oxygen is interacting with a hydrogen sticking off of the alpha amino nitrogen of the backbone somewhere else. So this is what we meant when we said non-contiguous amino acids. These amino acids are not next-door neighbors. Basically, we are twisting and bending the polypeptides so that we can get amino acids that are not real far apart, but not exactly close together. We can get them in close proximity, and we can use that hydrogen bonding to kind of, I don't want to say glue them together, but give a little bit of stickiness there. So like I said, what you're seeing here is called the alpha helix. You can see a kind of cartoony, not exactly realistic representation of it here on the left. And then you can see an actual three-dimensional space-filling model of a protein here. And you can actually kind of make out an alpha helix over here in this green area, and then kind of up here in this yellow area here. And then also, you can see it kind of all over the place. So a lot of proteins, once they fold into their final shape, they tend to have at least a few alpha helixes. So if you look at the alpha helix, and I like this picture which came from the Lotus textbook because it actually shows the side chains represented here. So this backs up what I was saying on the previous slide, that the side chains are not participating in the hydrogen bonds that hold this helix together. Like I said, the hydrogen bonds are between atoms that actually make up the backbone, not the side chains. So that's definitely good to remember. So one nice thing about alpha helices is that they tend to form in very predictable ways. So it tends to be such that you get a hydrogen bond between an amino acid that is 4 positions away from the next amino acid it is going to bond with. And you'll see what I mean here in a second. And then if you think of the alpha helix as kind of like a winding helical staircase, then every time you go a complete 360 degrees around the helix, you get exactly 3.6 amino acids. So two revolutions of the helix would give you 7.2 amino acids, and so on and so forth. So it's nice to know that alpha helices obey these rules that we've figured out here. As I said, the side chains protrude outward from the turns of the helix, which is nice because that actually means that since they protrude outward, they're still going to be capable of interacting with other side chains based on kind of how the protein positions this particular helix with regard to other structures within the protein. And then this is also a good exemplification of something that we talked about in the first video, the ability of the backbone to kind of bend itself into this winding helical shape really underscores the importance of that rotational freedom that we talked about earlier. You don't get any rotational freedom about the peptide bonds, but having rotational freedom with those other 2 bonds is enough to allow us to get secondary structures like what you see here. Okay, and then this picture down here shows you a octapeptide, so 8 amino acids, and you can just kind of look at this and identify what each amino acid is based on the identity of the side chain. So what you're seeing right here is aspartate. That's our first amino acid, so you can see its side chain right there. Tyrosine right here, you can see that phenolic ring with the hydroxyl group. Methionine, you can see the sulfur-containing functional group right there. Glycine, so the smallest amino acid, so it's kind of hidden right there. Tryptophan, you can see its ring structure right there. Another methionine here, another aspartate here, and then finally phenylalanine. So you're probably not to the point yet where you know exactly what the side chains are, but you might look at this and just try to get yourself to a point where you're like, okay, I get that this is this amino acid and this is this amino acid. Okay, so to help you out here, I've put these little red slashes in here to show you the position of the peptide bonds. So this is aspartate. Here's your peptide bond that links it to tyrosine. Here's your peptide bond that links it to methionine. Here's your peptide bond that links it to glycine, and so on. The purple dotted line here is meant to indicate if we were to kind of bend this octapeptide in such a way that we might get an alpha helix. This is obviously way too short of a peptide to get an alpha helix, but this is just to kind of give you the idea that alpha helices stabilize themselves by forming hydrogen bonds with amino acids that are at least exactly 4 positions away from each other. So we've got aspartate, we've got this carbonyl oxygen here. We're going to bend this peptide in such a way that we can get this carbonyl oxygen right here in close proximity with this amino nitrogen and its hydrogen right here. The hydrogen will attract that oxygen, and we should get a good hydrogen bond there. So one interesting thing that's talked about in the Lotus textbook is the idea that if you look straight down through an alpha helix and you actually take stock of which amino acids take up which position, a lot of times you may actually notice that hydrophilic side chains will often orient on one side of the helix, and hydrophobic side chains, which are in yellow here, the hydrophilic ones are in blue. The hydrophilic and the hydrophobic side chains tend to orient themselves on opposite ends of the helix. So this first figure, this set of 4 figures here labeled A, you'll notice that we kind of tend in most cases to have all the yellows on one side, which are hydrophobic, and all the blues on the other side, which are hydrophilic. This is nice because depending on what the final structure of the protein ends up being, we can actually position this alpha helix such that the blue side of it, which is hydrophilic, can protrude out towards the solvent and interact with the water, which is just what hydrophilic side chains would want to do. And on the other side, we can have the hydrophobic side chains, which don't like water, don't forget. They can be kind of hidden in the interior of the protein, hidden away from the water, which is obviously not something that we want to do. So when we talk about molecules that exhibit properties like this, in which they are simultaneously both hydrophobic and hydrophilic, the adjective we use to describe such things is the word amphipathic. So an amphipathic molecule or structure will have something that has hydrophilic properties and another part of it that has hydrophobic properties. And usually when that ends up being the case, it is highly, highly significant. Something practical later on in the semester when we start doing more involved labs, another kind of molecule that is amphipathic are detergents, which are very useful in disrupting cell membranes. Okay, so now let's talk about the other major type of secondary structure, which is called the beta sheet. Now, the beta sheet looks a little bit different than the alpha helix does, obviously, but the principle behind how this type of secondary structure gets formed and gets stabilized is exactly the same. Beta sheets are stabilized and formed by forming hydrogen bonds between oxygen and nitrogens of the backbone. Again, side chains are not participating here. The reason we get a different structure here than what we got with alpha helices is just because, okay, the amino acid sequence is a little bit different, and it makes maybe more sense to form a beta sheet here than it did to form an alpha helix. But like I said, it still applies here that we are using hydrogen bonds to stabilize this structure. So each beta sheet is going to consist of quote-unquote strands of about somewhere between 5 and 8 amino acids that are going to then fold back on themselves in one of two different ways. We can actually end up with both of these. We can either do this in parallel fashion, in which if you start with the N-terminus over here on the right, it goes this way towards the C-terminus, and then maybe, whoa, we have a whole bunch of different amino acids there, and then eventually we come back the other way, such that everything is oriented from N-terminus to C-terminus, N-terminus to C-terminus. Or we can do this in anti-parallel fashion, in which case the two parts of the beta sheet that are attracted to each other are pointed in opposite directions. They're parallel but pointed in opposite directions, kind of like two trains that are moving in opposite directions but on the same set of tracks. So in this case, you have the N-terminus here. We go from N-terminus to C-terminus, then we can bend back and come back the other way from N-terminus to C-terminus the other way. So here you can see a little bit, you can get a little bit of a better idea of what a beta sheet is going to look like. So these are sometimes also called pleated beta sheets because of the orientation of the atoms here. You end up with kind of up and down, kind of like it's a folded piece of construction paper like what you used to do back in elementary school. So what you're seeing here is an example of an anti-parallel beta sheet, and we can do this again and again and again until you end up with maybe something that looks like this for a protein. In blue, you can obviously see a couple of alpha helices, and then in green, you can see beta sheets that kind of wind back and forth as such. So those are our two major types of secondary structures. So which type of secondary structure you get in a particular protein is going to depend obviously on the amino acid sequence and on the primary sequence, but you should, in most cases, find a couple of examples of each within a given protein. So one whole protein may have several alpha helices and several beta sheets. The proportions are just going to depend on the protein. Okay, so something that is not strictly a secondary structure but is important in transitioning from one secondary structure to another is called a turn. So turns are U-shaped structures that usually consist of a very small number of residues, usually only about 3. And these turns are usually going to incorporate both proline and glycine, which we highlighted in the first video. You'll remember that proline has a very small amount of rotational freedom, so we said that you usually find prolines in places where the protein bends back on itself, so that's what we're talking about here. And then you tend to find glycine immediately after the proline. Because glycine is the smallest amino acid, it is able to, once you get that proline kind of turning the protein back, the glycine is able to help with that so that you can get the widest turn radius possible. So as was the case with alpha helices and beta sheets, turns are also going to be stabilized by hydrogen bonds, usually between, the backbones of the actual U-shaped structure here. So here you can see we have a hydrogen bond between the carbonyl oxygen of the first amino acid and then this nitrogen and hydrogen of whatever the fourth amino acid is meant to be here. So even though turns aren't strictly secondary structures, like secondary structures, they are stabilized by hydrogen bonding. So turns are important because they allow a protein to take on a much more compact shape so that their mass to volume ratio can be minimized and we can fit a much larger number of proteins inside the cell. Okay, so before we wrap up our discussion on secondary structures and move on to tertiary structure, let's talk about motifs and domains. So local secondary structures within a protein can actually combine together and come in close proximity to form actual biologically functional units, again called motifs or domains. So a motif can come together to form a very specific type of three-dimensional structure, and at this point we've obviously talked about how important shape and structure is, so that that part of the protein can interact with other molecules in very predictable ways. So the Lotus textbook only provides a couple of different examples, and there are really quite a few different examples, but we'll just go ahead and highlight the ones that the Lotus book mentioned. So one is called a coiled coil. This involves multiple alpha helices associating together, and this is usually going to be found in a protein that likes to multimerize, meaning that a particular protein likes to basically glom together with other proteins to form what you would call a complex that contains not just one protein, but several different proteins that all form one functional unit. And as we're going to find out a little bit later, that underlies how you get quaternary structures. Another example is what's called the helix loop helix, which produces a type of structure called an EF hand. One thing that helix loop helixes and EF hands tend to do is they tend to be able to bind calcium ions, which we are going to see on the next slide. And then finally, another type of motif is called the zinc finger. So what you're seeing here is that we form secondary structures in such a way that we orient cysteine and histidine residues together in such a way that they can coordinate zinc ions. Zinc finger motifs are very important because this is usually what you find in proteins that interact with and recognize specific DNA sequences. Usually we call these types of proteins transcription factors. Proteins that can enter into the nucleus, bind to the DNA, and stick these fingers into the double helix structure of the DNA, detect what kinds of nucleotides are there, and say, okay, this is the gene that I want to activate, or say, this is not the gene I want to activate, I need to go look somewhere else. So if you want to look at a particular example of a helix loop helix protein, look no further than the protein Calmodulin. So this is a protein that, as we said, has a very high affinity for calcium ions. But the problem is that calcium is not an ion that you're really going to find inside the cell most of the time. The cell actually goes to great efforts to make sure that the concentration of calcium in the cytoplasm is very, very, very low. But one thing we do is that the endoplasmic reticulum basically acts as a reservoir of calcium and can release that calcium into the cytoplasm in response to specific cell signaling events. So in the case where the endoplasmic reticulum would release a whole bunch of calcium, Calmodulin will bind to that calcium immediately, and this can lead to a number of downstream effects that is going to change cell and protein function. So I wouldn't sweat the details here, but this is just an example of how a very specific motif in a protein can participate heavily in a specific cellular or physiological mechanism. Okay, and then here's a three-dimensional look at the structure of an EF hand, specifically in Calmodulin. So what you're seeing here in the helix loop helix motif is that we're getting multiple alpha helices here together in such a way that you actually get hydrophilic side chains in between two helices oriented in such a way that you produce a space that is just right so that a calcium ion could fit in there. Other ions, like sodium, would be too small. Something like magnesium might be too big, so the distance between these two helices is just perfect to get calcium to bind in there. So again, if you weren't already convinced that the three-dimensional shape of the protein is what gives the protein its function, here's another example of that. All right, so now that we are done talking about secondary structures, let's go ahead and start talking about tertiary structures. So tertiary structure refers to protein folding that really goes a step beyond what we saw with secondary structures. So tertiary structure and folding is going to, this time, actually be facilitated by interactions between the side chains of the amino acids that made up the protein. You'll recall that secondary structure was all about hydrogen bonding among different parts of the polypeptide backbone. Now we can actually get the side chains involved here. So this is going to take us back to the discussion we had on the chemical properties of the different classes of side chains. So if you take that information, you should be able to come up with a logical way in which side chains can interact with each other. We can get polar side chains interacting with each other by either hydrogen bonding with each other, like what we saw in the secondary structure, or the polar side chains, if they are exposed to the outer surface of the protein, they can interact with the water that is surrounding the protein, which is what happens a lot. Acidic and basic side chains can interact with each other if they are in close proximity based on the electrostatic attraction we get between negatives and positives. Hydrophobic side chains are not going to be real gung-ho about being exposed to the water that surrounds the protein, so instead you're going to tend to find them buried in the core of the protein so they can be away from the water. And aromatic side chains, in addition to also being buried away from water, they have a tendency to kind of stack on top of each other, like what we saw. So the most significant driving force that actually gets you from secondary to tertiary structure, it's all about the hydrophilic and the hydrophobic side chains, specifically the hydrophobic ones. These are what we call hydrophobic interactions. Getting a protein folded into its final three-dimensional shape really is all about satisfying those hydrophobic side chains. You cannot underestimate the degree to which those guys do not want to see water. So the protein is going to try to fold in such a way that we make all of those babies happy, right? So poor baby hydrophobic side chain doesn't want to interact with water, so we're going to bend over backwards, literally, if we're the protein, to make sure that the poor baby doesn't have to see any water. Boo-hoo, right? So this, again, is what I meant when I said that the information hidden in the primary sequence is what is going to determine the final shape of the protein. So which amino acids we find in which order gives us our different secondary structures, and then will eventually get us to that one particular perfect configuration that makes all the side chains as happy as possible. Okay, so let's talk a little bit about the physics behind hydrophobic interactions. So we know at this point hydrophobic molecules, their deal is that they don't like water, right? Well, we need to explain that more than just kind of colloquialisms, right? So when a hydrophobic molecule is forced to interact with water, the water molecules are going to form highly organized cages around the hydrophobic substance. So look over here on the left. We've got a beaker of water. We've got two different orange hydrophobic molecules here. And if those two hydrophobic molecules are kept separate from each other, the water molecules are going to surround that molecule with as many water molecules as possible. So this is unusually organized for a biological system. And if you recall back to chemistry class and maybe biology class too, if you recall back to your discussion on entropy, This goes against the second law of thermodynamics, which says that all physical and chemical processes happen in such a way, usually such that the amount of disorder in the system goes up. Having such an organized system here, that produces a low state of entropy, which really goes against the second law of thermodynamics. So this helps to explain why hydrophobic molecules don't like water, because it goes against the laws of physics. Okay, so now look over here on the right. So instead of keeping those two hydrophobic molecules separate from each other, we instead allow them to glom together. This is going to reduce the number of water molecules that are necessary to form cages around the hydrophobic substances. This is less organized and therefore more disordered, and this increases the entropy of the system, which is much more preferred, again, according to the laws of thermodynamics. So hydrophobic interactions are all about getting those hydrophobic side chains in the protein to all huddle together. It's kind of like they're all getting together and forming a no water allowed club where only the hydrophobic molecules are allowed to join, and any hydrophilic substances like hydrophilic side chains or water itself, they are not allowed to be there. It's kind of like their own secret little club. The more hydrophobic side chains you can get interacting together, the higher the entropy of the system and the more favorable the protein intramolecular interactions is going to be. So you may remember this formula from one of your previous classes. Delta G equals delta H minus T delta S. So if you don't quite remember that, let me give you a little refresher here. Delta G is called the Gibbs free energy of the system. This basically is the end-all be-all for whether a particular chemical or biological process is favorable or not. We want this number to be a negative number. So a negative number is good and means that it is favorable, and the laws of physics say that that should be able to proceed. If it's a positive number, then it's probably not going to happen. It's unfavorable. So we want that to be a negative number. Delta H is called the enthalpy. So this is a little bit more complicated, but essentially enthalpy is just heat energy of the system. Again, we want this to be a negative number. So enthalpy isn't really that relevant here, but it's a part of the equation, so we want to go ahead and define it. T is the temperature in degrees Kelvin, so that's pretty self-explanatory. And then delta S is the change in entropy. We want this to be a positive number, so the more entropy and the more disorder, the more favorable the interaction is going to be. So you'll notice that if we do not have hydrophobic interactions, delta G and delta S are not favorable at all for us, so this is not something that is really going to be allowed to happen under the laws of thermodynamics. So if we try to do this, the laws of thermodynamics are basically going to say, I don't think so, and they're going to force us into a state that looks more like this, where we get hydrophobic interactions, delta G is a negative number, which is good, delta S is a positive number, which is good, so that's good all around. And this is going to help us explain why proteins fold in such a way that they do. When a protein goes about folding itself, it is searching for the folded configuration that produces the most negative delta G and the most positive delta S. If it tries to fold in such a way that has hydrophobic side chains interacting with water, that's no good, and it's going to keep folding until it finds that perfect configuration that is at the lowest energy state possible that satisfies as many of those side chains as humanly possible. Okay, so one thing that we mentioned in the first video that can kind of complete our discussion on tertiary structure here is the role that cysteine amino acids play here. We know because of those sulfhydryl groups that cysteines have in their side chains, they can form disulfide bonds, and this can help to further stabilize the folded structure of the protein. So this is more review than anything else, so we've already basically talked about that. So disulfide bonds can also be an important part of the formation of what we would call quaternary structures. So to be clear, the tertiary structure is the final three-dimensional shape of a single polypeptide. You don't talk about quaternary structures unless you're talking about a multimeric protein, a protein that consists of more than one subunit or more than one polypeptide. A good example of a multimeric, multi-protein complex that actually has a quaternary structure is something like hemoglobin, which you can see here. The actual quaternary structure of hemoglobin actually consists of 4 different polypeptides, which are called globins, 2 alpha globins and 2 beta globins. Each globin only has a tertiary structure, but when those 4 globins come together and form this quaternary structure, then you can go beyond the tertiary and start talking about quaternary. The Lotus textbook provides another example of quaternary structure by talking about the membrane protein called hemagglutinin, which you tend to find on certain types of viruses, such as the influenza virus. So here you can actually see part of the primary sequence of this hemagglutinin protein. So this is why I kind of encouraged you to try to learn the one-letter abbreviations for the amino acids. So what we have here is we have glutamate, alanine, leucine, glycine, glutamate, proline, and so on. So eventually you'll want to kind of get yourself to the point where looking at those is just kind of second nature. You can see the predicted secondary structures here. So we're kind of looking at an alpha helix here, beta sheet here, alpha helix here, beta sheet, alpha helix. And then if you actually look at the tertiary structure of this protein, you can see it right here. Again, we've got these alpha helices that we predicted. We've got these beta sheets that we predicted. You'll notice that it's stuck in the membrane, which some proteins tend to do, and we'll talk about that eventually in the class. So hemagglutinin does not exist as one polypeptide. It is actually what we call a homotrimer. Tri referring to the number 3, homo referring to the basically the idea of being the same. So a homotrimer is a quaternary protein that consists of 3 identical units of the same polypeptide. So basically, the virus is going to make or the cell that the virus infects, I should say, is going to make 3 copies of the hemagglutinin protein, and then those 3 proteins will come together to actually form the quaternary structure here. And then you can actually see the quaternary structure over here on the right, and it looks a lot more complicated. One other important example of the role that disulfide bonds play in the quaternary structure of proteins is in antibodies, and we'll have a good long discussion on antibodies later in the semester. But antibodies are these kind of Y-shaped molecules. They're also called immunoglobulins. So these are quaternary proteins that consist of 2 light chain polypeptides. That's what you see right here. They're called light chains because they're shorter. And then 2 heavy chain polypeptides. And you'll notice that the 2 heavy chains and then the heavy chain and the light chains are stuck together by disulfide bonds. You have a cysteine side chain here, a cysteine side chain here, cysteine here, cysteine here, cysteine, and so on. So we'll talk about antibodies much later on and how critically important they are to specific biotech applications, most of which involves the detection of specific proteins, but that'll be a discussion for another day, so let's not worry about it just now. Okay, so that was another fairly lengthy discussion, but hopefully you found it to be a good one. That's going to do it for our discussion on protein folding. Join us next time and we will start talking about the evolutionary relationships between proteins. See you next time.